Welcome to OrthoEpidemiology, Module 2, Data Sources and Characteristics. Last video we discussed experimental studies and observational studies. Observational studies still make up the lion's share of studies in orthopedic surgery, and in fact surgery as a whole. They are still extremely valuable because they allow hypothesis generation and allow you to study broader questions than randomized controlled trials. Observational studies can be thought of as getting a bird's eye view on a topic. Not seeing any one thing clearly, but getting a big picture idea of what's going on. A randomized trial allows for laser focus on a specific question and allows an investigator to answer with more clarity a very narrow question. These studies are, however, vulnerable to bias, confounding, and chance. This is doubly true for retrospective studies, where even the data source can be biased. As discussed in Module 1, there are 5 basic types of retrospective studies: case studies, case series, retrospective cohorts, case controls, and a grab bag of other studies such as ecologic studies, economic studies, decision analyses, and survey science. For each of these, a data source is needed, and selection of that data source is critically important to minimize bias contributed by the data source itself. In the previous video, we discussed the importance of a question in deciding on a design. After deciding on a design, the next question is where to find your data. Possible data sources include institutional data, multi-institutional data, clinical databases, administrative databases, pre-published literature, and publicly available data. Institutional data are the most common types of data used in clinical research. This type of data are collected through clinical care at one's own institution. The advantage of institutional data is the familiarity that clinicians have at the institution with the data and where to find it. This does cut down on missing data relative to the data obtained in other ways. In other data, no other recourse exists to find the data in other areas of the EMR. However, missing data are still common due to collection habits of the primary clinical data. These data are collected via clinical care. As such, if the researcher wants some data that wasn't warranted as part of clinical care, it won't exist. Additionally, if additional data points are desired, an amendment through the IRB is easily obtainable. Controls are also more easily obtainable using this type of data. Institutional data may not exist if the data were not needed as part of clinical care. As such, missing data points are common in the framework of a clinical study. As such, number of patients and hence statistical power may be an issue. Data quality are limited by what is needed for clinical care. Multi-institutional data can be a powerful answer to the lack of statistical power that single institution studies offer. Standardized definitions for data points are critically important in this regard to protect the internal validity of the study from misunderstandings in data collection. In this vein, subtle or sometimes not so subtle differences can be present in data collection. Additionally, missing data may not be recoverable, particularly when using large multi-institutional databases. There may be expertise bias if using a large database which is managed by several high-volume centers. This may decrease the external validity or generalizability of the study. For single-use studies using multi-center data, a data use agreement is needed between institutions. This can significantly hamper data acquisition and lead to increases in the time it takes to do a study. Databases are another option for obtaining larger amounts of data than available at a single institution. These can be used if no multi-institutional data are available or if that data would be very difficult to come by. The most important thing to remember when using this type of data is that you will need to play to the database's strength and remember its weaknesses. Using a database for a question it cannot answer would represent a fatal flaw in your research. There are 2 major types of databases: clinical and administrative. Clinical databases use data from actual patients designed and sometimes entered by clinicians for the purposes of recording clinical care. As such, for clinical questions, clinical databases provide superior quality research data in the framework of database research. NSQIP is an example of a clinical database. Data collection practices may vary from institution to institution, and as such, random error may be present in the data that are difficult to account for. Missing data and loss to follow-up are common. Study group data represents a hybrid between clinical database and multi-center data, and as such has some of the same strength and flaws as mentioned in both sections. Administrative databases are much larger stores of data. They are, however, of lower quality. Administrators enter the data for the purpose of billing or some other administrative purpose that may not be entirely clear. As such, unambiguous data points such as length of hospital stay, CPT coding, or mortality are generally acceptable to glean from these databases. Other data, such as clinical outcomes, are not as reliable. Some of these databases are population normalized, which makes them useful for abstracting population rates of a certain condition. Publicly available data are similar in scope to administrative databases, albeit with less granularity. They can typically provide basic information or population-level statistics. They are reasonably useful to make a quick and dirty assessment of incidence and prevalence, but they are often not robust or reliable enough for hypothesis testing for most purposes. NICE and KID, the online version, are examples of this. Previously published papers are an excellent way to see what is already known on a subject. Through the methodology of systematic review and meta-analysis, this data can be collated and written up to provide a reader with an excellent synopsis of the literature so far. They are second to none to pregame a study and to answer, is the study novel question within the finer rubric. As a standalone study, they are limited by the level of evidence of the member studies. As such, the lowest level of evidence in the member studies is the level of evidence of the systematic review. After deciding on the data source, inclusion and exclusion criteria must be assigned. Inclusion criteria determines the population of interest. In a statistical contest, inferential statistics obtained during your study will only apply to this population. Broader inclusion criteria will increase your external validity, whereas narrower criteria will likely help you detect a difference through decreased variance, but will narrow the scope of your results. In addition, too broad inclusion criteria may damage the internal validity of your question. For example, the Canadian clavicle study, in which patients were randomized to non-operative care versus plate fixation of clavicle fractures, included patients only 16 years old or older. If they had included children, it would broaden their generalizability, but multiple studies have shown no difference in treatments in this group. As such, the internal validity of the study could be questioned as they included a group which was clearly different from adults in their study. Exclusion criteria will prevent excess variance and exclude patients who are not of interest for the study question. If you wanted a cohort of patients who were exposed to a certain surgery, you would not randomly include patients who did not have that surgery and were not controls, as their inclusion would be meaningless. Additionally, you may exclude patients who are too different to be useful. For example, if you were trying to figure out what lab values were relevant when working up septic arthritis in a native knee, you might exclude patients who are on immunosuppressive drugs, as their lab values may differ too much from the general population to add too much statistical noise to your estimates. Step 5. Decide on data points. The two most important data points to decide on are the outcome of interest, in this case the y variable, and the outcome of interest, in this case the x variable. The diagram here contains a standard form of an equation of a line, which is the form that many research questions will take mathematically, in various forms or transformations. Other variables in this equation in retrospective research are the baseline risk of an outcome, indicated here as beta naught, and the confounding variables, which are the additional x's in the equation. It is reasonable to detail at this point what are the different types of data you may encounter and be interested in analyzing. Each of these types of data are analyzed differently and have different implications for analysis and interpretation. Nominal data. Nominal data has no inherent value. The advertising campaign Take the Pepsi Challenge was essentially asking individuals to choose between two items, Coke and Pepsi, that have no inherent value. Their alternative hypothesis would be that more people would choose Pepsi than Coke. Nominal data can come in binary form, as in yes or no, dead or alive, Democrat or Republican. These binary data are easy to recognize because it assumes only two possible values. As such, it is dichotomous, and it can be tested against a binomial distribution. It can be used also as a y or outcome variable in binary logistic regression. In this case, the outcome is the probability of the outcome being whatever is coded as 1 or a success. In terms of formatting your data, it is helpful to code your 1 or success as whatever you think will cause increased risk of the outcome if it is an independent variable or an input variable, because it will result in an odds ratio that are whole numbers and easier to interpret and easier for the reader to understand. Categorical data include nominal data with more than one possible outcome, like sports position, finger on a hand, or income level, assuming the income level is stratified into brackets. It takes the form of one of a limited and usually fixed number of values or categories. Chi-squared is a simple method of analyzing categorical data and tests the hypothesis that the number in a cell is independent of the category versus the alternative that they are related. In other words, the observed and expected frequency in each cell is not significantly different. Multivariate analysis is also available for categorical output. Ordinal data are a specific type of categorical data. Ordinal data implies that the data are ranked or ordered. This data may at first appear to be continuous because the data are generally numeric. It is important to make a distinction, however, due to its implications for analysis. Ordinal data has no inherent value or distance between values. As such, it is truly ranked categorical data. Two important questions need to be answered in regards to ordinal data when considering if it is ordinal or continuous for the purposes of analysis. Those questions can be understood in the framework of the pain scale, which is a prototypical ordinal data in surgery. The first question is my 2 the same as your 2? For the pain scale, the answer is clearly no, as it is subjective. The second question is someone in 4 pain in twice as much pain as someone in 2 pain? Theoretically, yes, but since pain is subjective and a ranking, the answer cannot always be yes. Continuous data, on the other hand, implies a full range of data with spatial relationship between numbers. My 38 Celsius temperature is the same as yours if measured similarly. A team with 20 points has twice as many points as a team with 10 points. As such, these are continuous variables and may be analyzed as such. For the purposes of analysis, data can be analyzed via parametric or distributional means, or nonparametric means. Parametric statistics are superior for making powerful inferences when the assumptions of the distribution are upheld. Nonparametric statistics involve ranking or ordering data and determining where in that order an observation is likely to be, or comparing distributions of data. They are considered to be less dangerous in the sense that they have fewer assumptions, but are considerably less powerful than parametric statistics when assumptions are upheld. Parametric tests are also called distributional. Assumptions are large sample size, which is typically greater than 20, and that the population follows a specific distribution, most commonly the Gaussian or normal distribution. Parametric tests allow for powerful inferences, but are not always appropriate. They can be misleading if assumptions are violated. For example, in a small sample, outliers will affect the results in a large way, and so if a sample is not strictly Gaussian or normal, the outlier will affect the mean in a way that could bias the hypothesis test, as in give a spuriously significant or non-significant p-value. Nonparametric tests have less assumptions and are less sensitive to outliers or odd distributions of data because they simply rank data. As such, the data set of 1, 2, 3, 4, 200 becomes 1, 2, 3, 4, 5 for the purposes of hypothesis testing. They are safer to use in small samples where the distribution is not strictly normal and where outlying observations are an issue. Normal distribution is pictured here. It is a typical bell-shaped curve, and a predictable percent of the observations will fall on either side. The larger a sample size, the more apparent a normal distribution will be in the dataset. Statistical tests exist to determine normalcy, but in most cases data can simply be plotted on or frequency tested to determine if the data are normally distributed. Skewed data are pictured here. This is a right skew, named so because the tail of the data are to the right. Because of the greater number of observations on the right, the mean will be shifted to the right of the actual most common observations, or the mode. The median is in between these two, the middle observation and the ordered data set. Other such datasets which differ from normal Gaussian or mesokurtic differ in something called kurtosis, which defines if a distribution is clustered closer or more distant from the mean. A leptokurtic dataset is clustered towards the mean, and a platokurtic dataset is spread out towards the edges more so than a normal distribution. If these differences are significant, it is safer to use nonparametric statistics for hypothesis testing. This is important to protect against biased estimates. This is particularly true if the sample size is small and there are outliers. Nonparametric tests have less assumptions. They do have an independent random sample assumption, which means that the data sample in theory should be a representative sample of the population you are drawing from. By way of example, consider the data set here to estimate blood loss with posterior spinal fusion with a data set of estimated blood loss in milliliters, including the data set 100 milliliters, 200 milliliters, 125 milliliters, 140 milliliters, 250 milliliters, 130 milliliters, 300 milliliters, 220 milliliters, and 3,000 milliliters. The mean of these observations is 496, and the median is 200. So this is clearly a skewed data set. So when deciding if this data set, forgetting for a minute about the large sample size assumption, would be appropriate for parametric or non-parametric statistics, consider whether you think the 10th value is more likely to be close to 200 or more likely to be close to 500 given the data you've already seen. Clearly the answer in this case would be closer to 200. As such, for this data set that is small with outliers, a non-parametric analysis would be more appropriate. Thank you, and please leave questions in the comments section.